The Life Cipriva project undertakes actions to conserve fish species that are only found in a few rivers in the Duero and Tagus basins. Until recently, these populations had been classified as belonging to other species within a wider distribution range, but detailed genetic analyses have revealed the unique, endemic nature of many of the fish species that inhabit these waters. One of the principal research teams involved in correctly classifying these endemic species has described many new species in Iberian waters. Carlos Marcos from the Duero River Basin Authority visited the director of this research team, Dr. Ignacio Duadrio, at his laboratory in the Natural History Museum of Madrid to find out more about the analysis being carried out at present and the main results obtained from detailed studies of biological samples collected during the Cipriva project. Ignacio Duadrio. We perform two types of genetic analysis. On the one hand, we analyze mitochondrial DNA, which is passed exclusively from mother to offspring, and this allows us to determine historical population changes, in other words, whether populations differentiated a relatively long time ago. On the other, we analyze nuclear DNA using some highly variable genetic markers that present frequent mutations over time to determine the genetic variability and separation of populations. The markers cover several things, enabling us to identify historical changes and population variability, the extent of these, and also genetic differentiation over a much shorter timescale. Mitochondrial DNA provides various insights into the fish inhabiting the Duero basin. The first of these is that some of the species became endemic to the Duero a very long time ago and were most probably isolated during the Miocene, which is approximately 10 million years ago. This is the case of the sarda, Achondrostoma salmantinum, and the Iberian spined loach, Cobitis calderoni. The Duero was really an endorheic basin, a vast lagoon where the Castilian meseta is today. In addition, we have found species with populations that have arisen within the Duero itself. How did this happen? Well, the Duero shift from an endorheic to an exorheic lagoon involved the formation of new units. For example, some rivers, such as the Huebra and Yeltes, were sited in high, relatively cut-off areas with a very small input of water that was insufficient to erode the Duero platform, and they have become isolated. In contrast, the Águeda River, which has a high input of water and a high erosive capacity, captures part of the headwaters of the Alagon River, and in turn the Alagon River captures part of the other Duero tributaries, and this process has favored the mixture of species and populations from both the Duero and the Tagus. For instance, we found that some western populations of the Bermejuela are slightly different from the eastern populations. The Esla Basin and the Terra became a unit long after these ancient endemic species developed. But what I really cannot stress enough is that the Duero Basin hosts a large number of species that are endemic to the Iberian Peninsula, the Duero, or a few basins, and these form a rich heritage that is very important to conserve.
Los monográficos del Cipriver presentan los aspectos más relevantes de este proyecto LIFE a través de una serie de reportajes en 4 capítulos de libre acceso a través del portal web de la Confederación Hidrográfica del Duero. Capítulo 1. Cría en cautividad. Capítulo 2. Actuaciones en cauces. Capítulo 3. Análisis y estudios filogenéticos. Capítulo 4. El Cipriver frente al cambio climático.